Right, well, ladies and gentlemen, Abdul is going to perform for you today a show in singing, dance, and sequence.
Yeah, I mean, where is my cool dancing partner? She's not here. She's going to get coffee. So we actually promised to dance. I think I'm just going to start, and I'm going to sit down.
Yeah.
You guys know who I am, and for the guys who don't know who I am, I'm heading the genomics course at the MRC Unit. And actually, people who've heard me speak before, I tried to get a line that's a little bit controversial, but I steal it from people like Moreno's line for the last time. This time I think the L'Oreal adverts, why am I here, is because I'm worth being here. So hopefully...
Because you're worth
it. Yes, I'm worth it. That's the one. Okay, so I'm going to talk to you about sequencing on the Nanopore platform. And I actually think it's quite apt for me to talk about joining Mark in this program, which I'm pleased that he allowed me to do this presentation. And also it's apt because actually I'm very, very excited about Nanopore sequencing and what Nanopore sequencing would do with the setup that we have here in the MRC in Gambia. And also alluding to what Joanne had said, that the units are going to invest a lot on genomics. I mean, with new building, data center, and hopefully a lot more machines to come in the next few months. So I'm excited about it. I'm also excited about this technology, how this goes forward. Okay, so nanopore sequencing. Charts, I think in 2010 are classified sequencing into 3 generations. So people who do Sanger sequencing is the first generation sequencing. So if you guys do the second generation sequencing, mostly what Anu and Mark had been talking about. So it's really good introduction to the fact is that those are what you call second generation sequencing. And even when Mark was alluding about the 1000 genome, there were 4 criteria in that. It's mean sequencing really the genome in a big piece rather than short pieces as we do in the second generation sequencing. So people are still quite excited about third generation sequencing. These are single molecules, so it means that you don't sequence, you don't need to amplify the sequence as what Arno had described. So if you're doing Illumina sequencing, because you're depending on fluorescence, you need to amplify each piece of DNA before you sequence. But with single molecule sequencing, each molecule is you're getting data from each sequence molecule as they go through. So there is no, if the technology works really well, you can think of working through the whole chromosome from one end to the other in one contiguous piece. And people are pushing, they're doing your protege, as we call it, Nick Loman and Josh Quick are going to hundreds of thousands of bases in one sequencing on a nanopore. So this is where they want to push the platform to go, so you can go huge contig sequence in one go instead of sequencing bits together and then patching it together and read. So that's why it's exciting. So I'm going to do Oxford Nanopore. It's not the only single molecule, as Mark also alluded again. Backpack Bio, it's a huge machine. You have to invest a lot to get a PacBio machine. It will fill half of this room. The computation for it is too. And it costs quite a lot. Even the smaller PacBio, it's a huge machine and it costs quite a lot. And for the setup like us, we can't invest in millions of quid worth of machinery because you need the support, you need service engineer will come in and do stuff. So really, we need to think out of the box a little bit. So I'm doing Oxford Nanopore, and Oxford Nanopore is the first one that lots of people have been talking about Nanopore for a while, even before the second generation sequencing, like what Andrew's been talking about. But Nanopore is the first one to realize that you can actually get machine out, and they've got MinION. It's been a few years. I got a MinION in my hand when they were doing the first map, what was called, they were telling you to give £1,000, you write a project, and they will send you one of the device. So I got one of those in 2014 or early 2014 when they came out, and you would run them. They would keep sending different kits, and you basically the experimenter because they were trying things with it. So they give you the device and they will send you flow cells. You need to send the flow cells back and so on. And recently they've launched Gridiron, which takes 5 flow cells instead of single flow cell. 5 flow cells, you can run them in one computer and do. And they also launched Prometheum, which is supposed to give you the same kind of data output as you get on a 10x. 10x cost 10 million pounds, so you buy 10 machines for $10 million and $10 million, and you can do the human genome for $1,000. So you have to invest 10 million to be able to do the human genome for $1,000. But the technology being sort of spread out, you can do it in ISIC 4000 so on. So those are, but I will concentrate probably mainly on the MinION. And next slide. I have a video to show, so I would escape out of this. Now this is interesting.
You can play within the page.
I can play within the page. Will I do that? Technology.
Where's your cursor?
It's next to you.
It's gone away.
It's there.
Where is it?
Okay. Just you see it.
Oh, there we are.
Okay.
No. Okay, I can't.
Or I can do it.
Yeah,
Okay.
you go out and then just play it.
Okay.
Oh, it's just a screenshot.
So how do I go out? Escape. Okay. Okay. So why do we want the sequence on the? It's alluded from the presentation that you can sequence DNA and also you can sequence actually RNA directly. So you don't need to convert because it's actually looking at a blockage of current. The RNA would have a different kind of current. So you can do RNA directly. You can do modified RNA. You can also do modified DNA. So you're thinking about you can sequence methylated stuff directly without having to do a modification and then modification before you actually do processing. There is no capital cost required. Okay, we paid $1,000 to get one of those devices. So we got 2 devices here. So said you can do ultra long read. It's portable, which is really the thing that excites me about it. And when I talk about some of the projects I'm thinking of doing here, that you will see how that fits in really well with that. They've got rapid prep, and I've got a slide on showing you if you use the transposon, the library prep can be quite short. I mean, really a few minutes. We've done it. It's not because what they said is something on hands, and that's how long it takes. High fidelity is something you use with enzyme. It means that you can slow down the pause enough to really tell between one base and another. So this is the word you use with polymerase, but it's the same. Large genome, and we're doing a cloud type workshop, and the cloud, what they've done, they've also invested quite a lot on the cloud part. It's free to use at the moment, but I'm sure in future we probably have to pay to use it. But for now, if you're getting Nanopore data, you can run it with in Metricore. You don't have to pay, and they store the data for as long as you want. So it's free at the moment. I don't know how that will change.
That's actually the problem. If you wanted to go Guinea-Bissau or somewhere where there's no Wi-Fi,
Yeah.
you can't use Nanopore, you can't analyze.
Well, so you
We
can.
are trying to do it where you can do it offline.
But you could do now. They've written it so that you can actually do it on a desktop manager. So you could do both. Yeah. Right. The device. It's if you want to come to the lab, we got 2. It's really small. It fits into this. And you can put the disposable part of the flow cells, which actually is the expensive. But if you turn the flow cell out, all the electronics are at the back of it. So in fact, that's why they want you to send every flow cell you use, you send it back, because they want to make sure that you haven't tried to rip the technology and sell it to Taiwan or North Korea. So you say every flow cells they sell you send back, and they check that you haven't done anything different to it. Otherwise, they will come after you and sue you. So we know now, people are able to generate 5 to 6 gigabytes of data. If you would listen to what Anu said yesterday about the different machine sizes, Mark had said, for the MySeq, you're probably looking at about 15 gigabytes around that. 15 gigabytes of data you get out of MySeq. So it's getting close to a MySeq range in terms of data. But really, we're talking about optimized libraries and all those done. So there's still an issue with the libraries because you need to get these tethered proteins that's going to pull your library through the things. If you don't get it right, you don't get the sequencing done. So these are proprietary DNA they've done stuff with, and people have actually got large amount of data. But in real life, the data size that we're getting is not approaching that. And it's still, because all the electronics on the machine, on the flow cells that they sell you, is still quite expensive, but they're trying to deal with that, where they would have sort of a carrier for the flow cell with the electronics, and the nanopore bits you can just throw away. That will cost you a few dollars. So that's coming. Smigion, they talk about it, that will come in the future. Two software that runs Oxford Nanopore. The Mino basically talks to the MinION, and then also that talks to the Metrical, which is the cloud. So you got Mino that's local. You can do base call locally, but you also can send data to Metrical, where you can actually do base call and also do some of the downstream analysis. There is a platform in Metrical which actually has quite a lot of workflow. Like if you're doing 16S, you really don't have to worry. It will do all the calls for you and do the classification as matches do. And it will also do something like what's in my pot. So if you doing with mixed sample bacteria, you can dump it into your flow cell, and it would do a quick dirty blast. I'll show you that hopefully in one of the I'll log on to Epi2M and show you some data, and it would just basically tell you what's in your pot. Okay. That I wouldn't show. It's about we're going to show you what the MinION does, but I don't need to. Couple of library preps. They're not as complicated as the Illumina one, but still, again, you need a couple of things where you have an adapter. The adapter you need to add. Also, you need to add a feather. Feather with a motor protein. You need feather brings the DNA towards the pore so that it will get the ratcheting to happen. So, and what would happen? Those will fall off. Once it's pushed the DNA through, those will fall off, and that DNA is done. You get a new one in that has got a motor protein, and it works like that. So the pores are, as just said, it lasts about 48 hours. So it's a protein. When people talk about nanopore, we always just talk about Oxford Nanopore. But people are working on graphene and silicon, where they're trying to use solid nanopore. That will come. The Japanese invest a lot on nanopore technology, so that at some point it wouldn't be using a protein. So because you're using a protein, it has a lifetime. So the lifetime, in fact, is that 48 hours. Just at some point, if you are collecting data, after a few hours, you can actually see few of your pores dying. They die in front of you, they go black. So you see green, and they keep disappearing, going black. Abdul has been doing this, sees that by the time you come 30 hours, most of your screen that were green, which are active, have become black because the pores have died in the time, because they're protein. So you're pushing currents through them to somehow they do that. But people who are investing on solid nanopores, it's probably the future in terms of longevity for analysis. 10 minutes rapid library prep does the same thing as the others, but you use a transposon. Illumina also use that for some of the rapid prep. So as Anu was explaining, there is also Illumina rapid prep. Again, because it's transposon, you need to test it against all organisms that you have, because it's looking for 9 bases. They might not be evenly spaced. If you look at the human genome, maybe that some of the transposon might be evenly spaced around your genome. If you're looking at some bacteria where maybe 80 reach or some kind of repetitive region, they might not be even spaced. So what you'll end up doing is that because you're transposing into your DNA, like punching holes, some areas you won't be able to punch holes, so you won't be able to probably be able to sequence that area. So, but because you do long read, if you even don't have small product, because you it's a problem with the Illumina platform. If you do the rapid transposon, sometimes you get areas that are not covered at all. We were talking to Susan about coverage. That's the kind of thing that would happen where some parts of your genome won't be covered because you use a transposon and it wasn't able to punch holes to make a library out of it. So that will happen. Is that clear? You can ask me a question. I can just rack it through this as I go.
I was just wondering about what the minimum input is in terms of nucleic acid.
So if you're not going to do any PCR, 1 microgram. Yeah. And people who are actually making cultures, that's nothing. People who are working with 1 or 2 cells or things that are really rare, it becomes then you have to use a PCR kind of to amplify and they'll run through. So you do still single cell, but you do kind of pre-amplification. But if you got tons of DNA, you don't have to amplify. You can dump that, make a library out of 1 micron. They're trying to push that down, a lot, but at the moment I think it works better with that kind of concentration. Okay, any more questions going forward? Shows you, go one back. Again, you put, there's a motor enzyme, there's a tether. It basically is like your lock zip and you open the zip and the single head of your zip end goes through the nanopore. And with the 2D, what it does is that you get much better information and also it reduces the error rate if you can have the same DNA piece where you push it one way, so you have one teeth of your zip and you turn it around and do the other teeth of your zip. So you can see that the accuracy will be increased because it's the same piece of DNA that's just gone through the one pore. So you can increase the accuracy by doing a 2D type of sequencing. So it's like paired end in a way. If you think of paired end, it's that kind of paired end because it's got 2 information for the one piece of DNA. Okay. Setup, very simple. This is a setup I had at the creek. Data comes live. You could see the green that you see tells you pores that are active, black ones do, and then you'll see this changing as you go. I mean, it changes as it will keep recording and as it's recording basis. And this is live information, and it tells you how many reads you've collected, what's the read length, and those are all done in real time as you're doing the analysis. I had 2 small projects I want to talk to you about. One I did at the creek. I mean, I think that's the thing about getting excited about being here. When I was at NIMR and at the creek, and I did a lot of sequencing, but I could tell you that 60% of the sequencing I did at the creek were either mouse or xenopus. So whether, because the developmental people into systems biology, they sequence quite a lot. So it's actually quite different. So one of the projects that we had was they were moving mouse from 3 different colonies, and they were going to move them to a new building at the creek. And because there's some of the immunology that they do that the microbiome matters to the immunology of the animal, I needed to sequence to make sure we know what the microbiome of the animals are before we move them. So the project was, could we do this? And we're able to design primers, use the Illumina system, and do 16S genomics on loads and loads of mouse poo. So I've moved from mouse poo at the creek, so this is a project. And also, when we did it with Illumina, and I wanted to test this, to look at the data that we get from the Illumina short read, and then and that's only doing the 16S if I do the structure as 9 variable region. And we're only looking at V3, V4 with the Illumina, so in fact, it means that if there is no variety in those, a lot of the reads get to unclassified because it hasn't seen those changes before. If you're able to do the whole 16S, you get more information. If you're able to do the whole genome, you get even more better information. So doing V3 and V4 is still very limited. You get 60-70% of your read goes on the unclassified. It's not because it's unclassified, it's just because you can't actually be able to, for the technology you have, you're not doing it. So doing the whole 16S, in fact, people are doing 16S and 23S all in one tandem on the Nanopore. I did just the 1500 thing and just look at the data between what you get with short read and what you get with doing the whole 16S gene. Did I get clear what I was trying to do? Okay. Tells you what I did. I mean, optimization, so this is a slide I did to give the talk at the end of it. we looked at, this is just showing two of the animal units that I looked at. Looks like quite similar. You expect it is a mouse poo, but the details is when we go back and look at saying what's the difference in the different bacteria or viral that load in the microbiome of the particular group units that I'm looking at. So those are details. Because it's a screening and more of a catalog. We don't know what we're looking at yet, but it's mostly is to know what we've got, and then we can go back and say we've got.
You compare that with the Illumina short reads.
Yes, yeah.
And
We get there is less unclassified with the longer doing the longer quiz.
but you get roughly similar proportions.
Similar to proportions, yeah.
There's no, but there are obviously biases in your primers. Sometimes
That-
leave out a taxon, downgrade it, and I guess that would vary whether you're using V3, V4,
Yeah.
or length. You have
Then you're using the long
biases.
Yeah. Questions?
Sorry, I might not understood. So in your comparison with the short reads,
Yeah.
the fragment that was amplified was still the whole 16x.
For the short read, it's only V3 and V4, 450 base pair out of the 1500.
Why can't the amplification steps be the same?
Because you're limited to, as Arnold alluded, you're actually limited to. So what you don't, you could basically, the other way you could do it, just think about short read, is that if you really want to do it, if you want to do it cheaper in the Illumina, if you really want to do it cleverly, you can actually do all the region amplified and then concatenate them and put it together. For the moment, 450 works with the MySeq kit because you got 600 base pair that you can sequence. So you basically sequencing those in full. So if you were doing a longer read length, you would have to break it under. So there is no alignment. There's an alignment, but there is no, what's it called? When you put contigs together, is you're only looking at one smaller bit. But actually people are doing where they don't just do V3 and V4, they do several PCR and dump them all into one pot and do. But what he does is that the bigger regions that you do, the less samples you can dump into your, because you've only got so many reads to play with. So you could do 384 samples maybe with this 16S. If you want to do all the 1500, you might have to reduce that as a different amount. So it's cost.
With the short read,
Yeah.
you can't really call down to species level. You can just about maybe even genus level.
Yeah.
A bit dodgy.
Yeah.
Whereas with the whole full length, you'll get
A little
all
bit
the
of
information you can. Right?
Is that okay? Can I go on?
So what about the error rate?
So it's a problem, but I think that's because one of the things that you will lose a lot of data because the error rate is if the error rate is high and it cannot line them, it will throw it away and you become probably unclassified. So maybe what you need to go back is to look at those unclassifiers and say, is it because actually the Q scores were really poor, so you can't put in a bin, or is it because it's an organism you can't find? So that, but you do get a bit more out of this than you do. Okay, so here we have I said most of the stuff that I'm doing with Nanopore was a collaboration with the Crick. They gave Crick and also the people at Nanopore. So all the stuff we've done and the kits that I've brought here, MRCG haven't paid a penny. So everything I brought with me. So I bought the 2 Nanopore, the computers they let me have, and actually got servers that Archie knows they also let me have. So somehow they were happy to just give me stuff and let me go. So they don't want me back. So I think. Okay, so also, I was accepted again for the VIP. That's the only thing that MRCG has paid for. We got Voltrax. There is no kit yet for the Voltrax. So that would take away some of the differences we have with the library prep, because an automated library prep is also a handheld device. So that really will be quite exciting to take that in the field somewhere where the library prep is standardized and your sequencing will be standardized, provided you can throw your data away or you can do it locally. Okay. So at the MRC, there's a lot of things we can do here. And so that really excites me about using this technology. I mean, when you're thinking about monitoring infectious diseases and or you're looking at outbreak, and I will come back to the one small project that I did with Martin if he was here, and you're looking at about outbreak, if you can actually go, and one of the things that happened, and it's really nice that Eben is here, there's an issue about when you're trying to collect sample on the field, trying to do microbiology, so you really go through a huge risk of getting people that are experts to the field. They get samples, they collect, they try to grow. There's an issue about the infrastructure where what they meet in terms of how they will store samples. Then there's also an issue about how do you bring those samples to Gambia, where you got your technology, or somewhere in Europe you get your technology. It's a huge task, and I wouldn't say what the pitfalls of it. Maybe Eben over lunch will tell you what issues they are. But actually, it fitted really well with my time here because I've turned up and they got some issues with some samples, and they said, but could you sequence it on the Nanopore? So the next time I've been told that when there's an outbreak, I would have to take my Nanopore with me to wherever they are. So we're collecting sample, we'll try to see if we can actually sequence them side. Because if I give you the catalog of issues that he had, which, I mean, he had an afro before when I met him, now he's lost his hair. Okay, so one project is to see whether we can look at from a CSF of samples they collected from an outbreak sample, a real outbreak sample, whether we could sequence it pre-cultured and post-culturing. Okay.
So do you know, that's what I was getting at with in terms of your minimum input. So for samples like CSF, do you know that you're getting up DNA, and a lot of it will be host? But,
Yeah, and I would allude to the data for the CSF. So CSF is an issue still because you need to do human depletion. People that Mark's collaborating with, I think Eben's other half, they're actually doing as a clinical setup where they got in human clinical sample looking for bacterial infections, and they're able to do it by doing kind of clever depletion. So then they will make, so what you do is, it's actually also in Illumina. Then you think about it, what your input material is and what you sequence. So, and this is quite a logic thing to think about again. So I would ask you for 200 nanogram to make a library, but I'm only going to load fentanyl sulfates on my machine. But I can't ask you for fentanyl because I will not be able to make a library on fentanyl sample. So I'm asking you for a lot of sample for what I actually need to load on the machine. So you could think logically that if you can make a library and you deplete and you only get your host that you can, the thing that you want to look at, that would be a clever way to do it.
But for the mina and the loading is not 1 micron.
So it's less. If you lose, you lose a lot. I'm trying to work out in numbers, I can tell you, but it's a lot less. You think in the pore is only, if you think of a number of molecules that you get in Avogadro's number, then divide, so, and you look at the space in the pore, really it's a nano, it's still a microchip. So you can work out the volume, work out numbers, it would be very, very little. Make sense? And actually, also you can clever things that you could do. You can look at data from a particular pore and close it or take the feather off. So there's another clever way that you do. If you put a mixed samples and some of our humans, you can just say every time you see human, can you just take it off? Because once you take it off, they cannot be sequenced again. Does that make sense? So you can always just preferably waiting for the bacterial sequence to go through. It's a clever way you can do it. And people are doing that. Okay. Data for some of the samples from we did isolates, which means bacteria that Evans grow and they just mainly bacterial cultures. And also we also look at CSF. CSF, as again, was challenging without doing the reduction. I won't show you the data, it's quite difficult to get, but if you do the culture, you can easily serotype what you have.
If you do the reduction, you have enough DNA.
You have enough DNA, but still there's still an issue that it's not good enough because when we did the reduction, and we need to go back and see, you're getting other species, but they're not human, but Clostridium, some other fungus, something else, which could be probably been abandoned in a human sequence, and we just basically, you do it because you break and you take a human sequence out. But if you actually add a viral sequence, they might just come from the human. They're nothing to do with your sample, and you still get it. And that makes sense in thinking. Makes sense to some thinking. Outcome. We've sequenced, and I was going to show you EP2M. I will show you how much time have I used. My time's done. Yeah. So I will not, but I can show you data.
Mark is not here, so you can.
I can carry, and I will show you the data on one of these. But what it is that we managed to sequence some abli, not me, sequence some isolates, and able to at least classify it as strep and some class of strep. And I could show you that before, if Mark doesn't come in. Only one thing is that people are excited in the unit, as I'm excited, and it's just to thank my team. They're all here, which I'm really pleased about. So it shows there is a little collaboration. And also, even before the course started, and Mark's team had gone and spent some time, so which I really was happy about. They gone and spent time with them so that they at least can see whether we're doing the right thing and asking the right question going forward. And also was very, very happy that all of them wanted to be here. So my team, Pender, on the corner, Jara and Ablai, and work on the Minion mostly done by Ablai. Okay, that's me. That's Max here. Thank you.
Good.
Of that.